We'll be starting with Markus Harrigan, who is an MD PhD student from Ohio State. He completed his PhD in investigating muscle wasting after spinal cord injury, funded by an F-31 training grant. He is completing his training at Ohio State University and has done a really nice job with us over the last 3.5 weeks. Looking forward to your talk, Markus.
Great, I'll just share my screen really quickly. All right, good morning, everyone. First off, just wanted to thank the department for the opportunity to present my research as well as also rotate here. It's been a great experience so far. So today I'm going to be talking about muscle wasting after spinal cord injury. So traumatic spinal cord injury, all of you are familiar with this, but obviously you can get an external force, usually a vertebral body that can impact the spinal cord, and the result is irreversible injury of motor, sensory, and autonomic neurons. Muscle paralysis is the most prominent consequence of this, but today I'm going to talk about how this might not just be related to motor neuron injury. So we know that paralyzed muscle is rapidly affected by spinal cord injury. And we know that skeletal muscle atrophy is endemic in the spinal cord injury population. We've seen that paralyzed muscle size can decrease by 50% already within 3 weeks. And actually the majority of this loss occurs within the first week to 10 days after injury. And this is demonstrated really nicely here in an MRI where above you see a control patient and then below you see a patient about 6 weeks after injury, and you can see how that muscle size is rapidly decreasing. So our question was, could therapeutically protecting muscle improve spinal cord injury outcomes? So your question might be, why should we focus on muscle though? And why should we study muscle after spinal cord injury? So we know that severe muscle atrophy occurs early after injury and that it reduces survival, impairs rehabilitation, and it facilitates the development of secondary complications. And there also aren't any available clinical interventions to prevent muscle wasting currently. So what do we know so far about what causes muscle mass early after SCI? The ongoing paradigm for decades has been that muscle atrophy after spinal cord injury is just related to disuse, not using the muscles. If you don't use it, you lose it. So we're considering a different hypothesis because in reality, we know that spinal cord injury causes rapid and severe paralyzed muscle wasting beyond just what disuse could explain or even just acute trauma. You don't even see this rapid of muscle wasting in patients that are in the ICU without a spinal cord injury. And we also know that also fully innervated non-paralyzed muscles also rapidly atrophy. So these are muscles that are fully functional, are being used, and those are still wasting away. And we see a 30% reduction early after paraplegic spinal cord injury in those upper extremity muscles. So to investigate a new paradigm, so investigating a new paradigm here, I'm just illustrating it graphically. So if we think about paraplegic spinal cord injury, and I'm not sure if you can see my mouse, but if we do a thoracic injury, obviously you get paralysis of the lower extremities and you have fully functional upper extremities. And so this is depicted here with this regional muscle paralysis of the lower extremities in purple. And then the existing paradigm, which is that then you have atrophy of these lower extremities, but preservation of the muscles above the lesion. But instead, we're suggesting that this regional muscle paralysis actually still results in systemic muscle wasting. So our goal is to identify targets which could be targeted during this critical period between injury and rehabilitation to protect muscle. So our experimental model, similar to the human, in mice, you can also induce paraplegic spinal cord injury. Here we looked at 2 different levels of spinal cord injury within the thoracic cord. T3 transection and T9 transection. Here the hind limbs are paralyzed and the forelimbs are non-paralyzed. So by comparing 2 different levels of injury, both of these only cause hind limb upper motor neuron injury. So they both only cause hind limb paralysis, leaving the forelimb fully intact. And without changing this motor innervations or paralysis, we can compare lesion level dependent effects. So by comparing the high thoracic injury to low thoracic injury, we can look for mechanisms that might be propagated from the spinal cord itself without changing paralysis status. So we really honed in on the triceps brachii and the forelimb as an index muscle of potential systemic effects of muscle wasting, as opposed to the gastrocnemius and the hind limb, which would be subject to both local effects like paralysis, disuse, and the systemic effects. And so what we did was we originally looked at just body composition over time, really honing in here on lean weight. And we can see that already within 1 week after injury in these T3 SCI animals, which will be read throughout the rest of the presentation. So this is the high thoracic injury relative to the low thoracic injury in gray, and then the sham animals in black. We see that within 1 week already, these high thoracic injured animals have lost about 20% of their lean mass, which mostly is comprised of skeletal muscle. And then to figure out where exactly lean mass was being lost, we harvested our index muscles above and below the lesion. And we saw that already at 3 and 7 days after injury, that both the paralyzed muscle and the non-paralyzed muscle were rapidly reduced in a lesion level dependent manner. So they were more severely reduced after the high thoracic injury, the T3 injury. So in brief, we identified a spinal cord injury lesion level dependent severity of acute systemic muscle wasting, suggesting that there were mechanisms originating in the thoracic spinal cord injury, which are spinal cord injury level dependent, and exacerbate systemic muscle wasting during acute T3 spinal cord injury. So then our question was, what could be the catabolic stimulus that's causing all of this? And so first we just looked at basic H&E histology of the muscle, which really didn't clue us into anything, was pretty non-specific. We also dug a little bit deeper and we looked at the fiber composition of the muscles. So the fibers can tell you a little bit about what's going on, but these were also non-specific findings that really just told us that there was something systemically affecting the non-paralyzed muscle, but we didn't necessarily know what. So when you have muscle atrophy and there's a lot of different things that can cause catabolism. Here we see, for example, denervation, disuse, inflammation, infection. We started off trying to rule a few of these things out. We made sure that there were no differences in eating, no differences in liquid intake, things like that. But we realized it was going to be a pretty tall task to rule all of those out individually. The nice thing is that all of these potential mechanisms do have molecular signatures, as illustrated here on the right, just as a general overview. And so our question then was, could we look at the molecular signatures to potentially identify what might be happening or what might be the trigger upstream? So we turned to RNA sequencing, and here you can see the volcano plot and the heat maps, but I'm not going to get into the nitty gritty of that. Really what we ran was an upstream regulator analysis, which really clued us into a specific candidate catabolic mechanism. So what we did was we compared the T3 after T9 gene expression profiles, and there were lots of differentially expressed genes, but this analysis was able to tell us what potential molecule upstream could explain the molecular signaling differences that we're seeing in the muscles of those T3 versus T9 animals. And what that analysis told us was that glucocorticoids were the most likely upstream molecule causing these differences, as illustrated here. So in the muscle of T3 spinal cord injury animals, you can have glucocorticoids bind to the glucocorticoid receptor in the muscle, cause transcription, and ultimately we hypothesized result in systemic muscle wasting based off of this analysis. So then our next question was, are glucocorticoids potentially increased after high relative to low thoracic spinal cord injury, so hypercortisolism? It's important to note here that the adrenal gland receives most of its innervation above the T9 thoracic level, at least in the mouse, versus with a T3 injury, you actually cut off the innervation to the adrenal gland. And so we did find, in fact, that after T3 spinal cord injury, corticosterone or glucocorticoid levels were more significantly elevated early after injury. So that T3 spinal cord injury disinhibited the adrenal glands, causing a hypercortisolism. And that's illustrated nicely here by the expansion of the adrenal cortex in those T3 spinal cord injured animals. So our evolved hypothesis was that this concurrent spinal cord injury level dependent hypercortisolism was likely driving the spinal cord injury level dependent systemic muscle wasting. And we wondered whether protecting the muscle from acute hypercortisolism could diminish wasting early after injury. So we turned to 3 separate interventions. The first one that we did was adrenalectomy. So we just removed the adrenal glands entirely, which are the endogenous source of glucocorticoids in the mouse. And we looked at the same outcome measures that we used when we were characterizing this phenotype. And so we saw improvements in lean weight. If you just compare the red to the white bars, so we saw improvements in lean weight, triceps brachii, so non-paralyzed muscle mass, and paralyzed gastrocnemius muscle mass. So we saw improvements in all of our outcome measures with adrenalectomy. So ablation of the adrenal hormones, which we hypothesized glucocorticoids was protecting muscle mass. So next we went to mifepristone, which is a pharmacological glucocorticoid receptor antagonist. And here we were blocking glucocorticoid receptors with this antagonist and observing the effect on our muscle outcome measures. And here we also saw improvements in lean weight and improvements in non-paralyzed muscle mass. So we were blocking glucocorticoid activity. Here we can't say that we were specifically blocking it at the muscle glucocorticoid receptors. We were blocking glucocorticoid receptors systemically. So our next step to really hit home our hypothesis was we turned to a skeletal muscle glucocorticoid receptor knockout mouse model. So here what we were doing is we specifically knocked out deleted the glucocorticoid receptors in the skeletal muscle and nowhere else. And we were able to do this relatively close to the time of injury, so 2 weeks before the injury. And here we've demonstrated that that the glucocorticoid receptor expression is reduced in the knockout animals relative to the control animals in both the forelimb and the hind limb. And once again, we see improvements in lean weight, non-paralyzed muscle mass, and paralyzed gastrocnemius mass. And most importantly, if we compare the T3 injured animals to the T9 injured animals, we see that there's no longer a lesion level dependent difference in any of these parameters. So essentially we're able to ablate the spinal cord injury lesion level dependent severity of systemic muscle wasting by targeting glucocorticoids through these 3 interventions. So in summary, we're suggesting that after high T3 spinal cord injury, there's hypercortisolism that results from disinhibition of the adrenal glands. This glucocorticoid excess then causes a systemic muscle wasting of both non-paralyzed and paralyzed muscles at an early clinically relevant time point. And it's a systemic mechanism that originates in the spinal cord after T3, but not T9 SCI. That was it. I want to thank my lab, the funding sources, also once again, the opportunity to rotate here and present this research. I'm happy to answer any questions that anybody has.
Thank you, Markus. Outstanding. I think we have Jenny Zhou with us, and I would love to hear if she has any comments about your work.
Yeah, nice work. So this really sort of highlights the systemic view. Not spinal cord injury not only affects the the neuronal circuits, but also affects, I think we were just at the Gordon conference in Italy together with Markus. I think the lab at Ohio State also talked about liver, liver problem after spinal cord injury, and and this talk is really nice to highlight the muscle atrophy. So my question is, have you guys looked at the in clinical? Obviously, we have a lot of spinal cord injury, actual patients. Have you guys thought about measuring the steroid level in actual patient population?
Yeah, my PI has been working on that. So interestingly, this phenomenon, this hypercortisolism that seems to be lesion level dependent, is also involved in spinal cord injury immune deficiency syndrome. So they've been doing that actually for a few years at this point, looking at glucocorticoid levels, also catecholamine levels, a whole host of hormones in patients, at varying time points after spinal cord injury, and with various levels of injury. Nice. And that work is based in Berlin.
Yeah, do they see a correlation of the the level of the injury versus the level of the glucosteroid? Yeah.
I believe that's unpublished, but to my knowledge, yes.
Yeah, nice. Thank you.
Yeah.
Hey, Markus, I'm sure that if you put an Apple Watch on a patient who had a spinal cord injury, that there'd be less activity in the whole body just because you're laying in bed. Even in the upper extremities, there's going to be less activity. Do you think that that contributes?
I think it's probably a component of it. I think the other interesting thing is that glucocorticoids can actually synergistically increase disuse atrophy. So it could be that these are, things that are going on simultaneously and actually encouraging each other as well. Interestingly, in rodents, I know there's some work at UCLA by Reggie Egerton where even very small amounts of muscle movement, just for a few very minimal amounts of time throughout the day seem to be enough to hold off disuse atrophy or keep it at bay. But I definitely think that it's part of it. And we're not saying that glucocorticoids are driving all of the muscle atrophy that we see after spinal cord injury, but we think specifically after high level injuries that this is a major contributor to it. But certainly the disuse is at play as well.
Great. That was a great presentation. Thank you.
Thank you. Thank you.
Thank you, Markus. Our next presenter is Momin Mohis, who's also been with us for 3.5 weeks. He completed his undergraduate degree at Johns Hopkins in biomedical engineering and economics before moving on to the University of Wisconsin School of Medicine, where he's in the urban medicine track at the Milwaukee campus. He's going to be presenting today on the subject of pediatric deformity surgery and predictors of short and long-term morbidity.